Mouse has like a. Because it's like trying to go Wi-Fi over that thing. You're up.
The downtime is nice.
All right, good morning, everyone. Go ahead and get started. Just real quick, our next meeting will be in November, so good luck with harvest, everybody. The meeting date will be November 19th, and we're putting together a spotted lantern clock panel. Eric Kohler has a pretty robust panel, so we're having Julie Urban and Brian Walsh both come out from Penn State to present to us. Then we have Francesc Marco, postdoctoral scholar at UC Riverside, will be here, and Andrew Klein from CDFA. So I texted Eric. I don't have the exact details on the hours. It'll either be 2 hours or a full half day presentation to give everybody enough time to do their thoughts. And then we'll be rolling into 2025 season. Today, we're pleased to have Dr. Cristina Lazcano with us, and she'll be presenting on evaluating her research on evaluating the benefits of no-till and regenerative management for vineyard soil health. And so real quick, Dr. Cristina Lazcano is a soil ecologist and associate professor at the Department of Land, Air, and Water Resources, University of California, Davis. Her international career has taken her from Spain, where she completed her PhD, to the Technical University of Denmark, University of Calgary, Canada, and UC Davis. Before joining the faculty at UC Davis in 2019, Dr. Lazcano was an associate professor of soil ecology at California Polytechnical State University, San Luis Obispo. Dr. Lazcano currently leads a team of 20 researchers at different career stages from different backgrounds focused on the understanding of the links between soil biodiversity, agroecosystem functioning, and human health. Dr. Lazcano's work identifies the soil ecological interactions responsible for carbon sequestration, nutrient availability and retention, and agroecosystems, and how they can be managed to increase soil health and sustainability of agricultural production. She's also worked with crops like corn, tomatoes, strawberries, and for the last 6 years, her research is focused on wine grapes. We're grateful for that. So through on farm collaborative research, Dr. Lazcano hopes to help wine grape producers understand how to improve soil health, environmental quality, and agroecosystem resilience while producing high-quality grapes. Super excited to have her here. Her recent work on no-till vineyards was awarded with the 2023 Viticultural Paper of the Year Award from the Australian Society of Viticulture and Enology. Congratulations on that. And thanks for being here.
Yeah, thank you for inviting me. Well, it's really exciting to be here. Thank you. And thank you for being here. I know it's getting really busy at this time, so with harvest and that. So yeah, I'm going to be focusing on our work on no-till. This is something that started 8 years ago, and it has been done with lots of collaborators. Part of them are Cal Poly at UC Davis, USDA. So this is the work of a lot of people involved in this, what I'm going to be showing. And I want to talk about the benefits of no-till from the perspective of climate change mitigation and adaptation, which is one of like the concerns at the moment of the agriculture industry. There are many other aspects we can discuss as well. And so, okay, let's see if this works. All right, we tried it before, but oh yeah, it's just the wrong arrow. I got it now. All right, so what we're seeing now, it's like one of the biggest concerns is uncertainty in the climate, right? And climate, what we call climate whiplash, which is like the change between extremes. And so the dry years are getting more dry, and that affects, you know, the amount of water we have for the growing season. And then the wet years are getting more wet, and then we have floodings, right? Which is also a whole complete different set of challenges for the vineyards. So this is on one of the challenges. Then obviously, as we are already experiencing, at least, I mean, if you live in Sacramento like I do, like, you know what heat is, and it's getting hot. Our temperature has been increasing globally, especially over the last decade. So this is what you see here. You can actually go to this website, showyourstripes.org, and then kind of download this image for your region of the world. This is for San Francisco, it's the closest I could get to here. And it shows average temperatures with respect to the 30-year average. And so the blue colors indicate temperatures that are cooler than the average, and the red colors are temperatures that are hotter, right? This is from 1860 all the way to the present. So it is pretty obvious that average temperature has been increasing, especially, as I mentioned, over the last decade. And there's also very strong evidence that says that this is because we are increasing the concentration of gases in our atmosphere, CO2 mostly, but also other greenhouse gases, right, over the last years as well. So the IPCC, which is the Intergovernmental Panel on Climate Change, has released a report in 2022. This was, I guess, the most recent, which says that if we want to reduce the increase in temperature to 1.5 degrees Celsius, we need to take urgent and drastic action. So this is a reality. We are already in the middle of it. Temperature has increased, and we need to just do something about it. And that means not just reducing emissions, which is mostly the use of fossil fuels, but we need to think about how to capture the carbon, which is already in the atmosphere, right? So the two things need to be done in order to avoid increasing our temperature more. So what does soil have to do with all of this? You probably know that soils hold a lot of carbon in them, more than the biosphere and the atmosphere combined. There's more carbon in the soil than above ground. And that is because there is an input constantly of the carbon from plant biomass, right? That plant biomass is decomposed, turned into nutrients. Those nutrients allow the plants to keep growing, right? So this cycle of photosynthesis and decomposition is what keeps the carbon flowing from the atmosphere through the soil, and then eventually makes up for the soil carbon and nitrogen, right? So this is the mechanism in which naturally the CO2 can be captured, but we have to keep in mind that at the same time that we have that capture, naturally this release of other gases, CO2, but also nitrous oxide and methane, which are the products of some of the decomposition processes that happen in the soil. And that's natural, right? But everything should be in a balance, and overall, the CO2 cycling through the soil year after year, it leads to a net gain. You have to think about this as you think about a bank account. If you want to save money, your input, right? The amount of money that goes into your account has to be more than the one that goes out. And that's what happens with the carbon in the soil. And then overall in the planet, the best mechanism to bring carbon to the soil, the major source of carbon, as I mentioned, are plants, right? Animal sources are also a possibility. That was so animal sources through, for example, manure inputs, but those are minimal compared to what the plants bring to the soil, right? And when we think about plants, it's not just what we see above ground, which contributes to carbon a lot, but also what happens below ground. And this is a drawing that I like because it kind of shows a diversity of plant species that can exist naturally in prairie systems. And it highlights that for most of these species, the majority of the biomass is actually underground. It's in the roots. So we have to think about that as a very important source of carbon as well. So when the plant dies, that litter, either from above or below ground, will contribute eventually to soil carbon. But also when the plants are alive, they are constantly producing carbon. That's what some people call liquid carbon. It's carbon that is produced as root exudates. So, think about like the roots are sweating all the time and releasing tiny droplets of carbon that the microorganisms are going to use as food sources. And, so that carbon is transferred from the atmosphere to the plant and eventually to the microbes in the soil, which grow, right? So what you're doing is growing microbes through the plants. Okay, and so we want to put carbon in the soil, yes, to mitigate climate change, but we also want that carbon to stay. So here's where the question of permanence, it becomes important. When we talk about carbon sequestration, we're talking about that carbon remaining in the soil for a long period of time, at least more than the CO2 lingers in the atmosphere, right? So we want that carbon to stay. And from that perspective, from the perspective of the permanence, when we think about soil carbon or soil organic matter, we think about it in two main fractions. What we call the POM, or the particulate organic matter, and what we call the MEOM, or mineral associated organic matter. So the particulate organic matter is your typical plant litter after it's in the ground. It can be fragmented, translocated by the fauna, like beetles, earthworms. Those little critters are gonna take it, break it down a little bit, chop it down for the microbes to finish the job, right? So that fragmented litter that hasn't been completely decomposed is what we call POM. The permanence time of that organic matter in the soil is between 1 to 50 years. So it's relatively low, right? This is important for the soil to function. It's a source of energy. It's going to be used by microbes, but it doesn't really lead to carbon sequestration. What we want is for that particulate organic matter to be finished by the microbes, to be completely decomposed, turn into microbial biomass, and then eventually stick to the minerals in the soil. Why? Because once the carbon is trapped in the minerals, it's not accessible for further microbial decomposition, and that makes it stable. And we're talking about 10 to 1,000 years, right? So this is one of the most important mechanisms of carbon stabilization and carbon sequestration, right? Most of the times, when we talk about carbon in the soil, the emphasis is put on total carbon, and that's important, but we also need to think about the carbon being stable, and for that we need an active microbiome, and we need like the whole soil food web to do that type of work, to stabilize it, right? Okay. And while we can sequester carbon in the soil and mitigate climate change, I mentioned before that we can also release greenhouse gases, and some of them are dangerous, like nitrous oxide or N2O. It's dangerous because it's 273 times more powerful than CO2 in global warming. So it has a higher global warming potential than CO2, and we need to be very careful about this gas, right? This gas is released in relatively high amounts from cropping systems, and it's produced during two processes. The process of nitrification, you know this as the oxidation of ammonium into nitrate, and then the process of denitrification, which is the reduction of the nitrate all the way to N2, which is a form of atmospheric nitrogen, okay? The interesting thing is that these two processes happen under different conditions. Nitrification is an oxidation process, so it needs oxygen, so that means it needs to be a well-aerated soil. Denitrification happens under anaerobic conditions, so when there's not enough oxygen, and that happens usually when soils are water saturated, right? So when you have a lot of water in the soil, you have higher denitrification rates, and if you, in addition, have a lot of inorganic nitrogen just laying around without being used, it's gonna be released, right? So you have to think about those two things, having a lot of nitrogen in the soil and maybe having water saturation, and that will lead to high peaks of nitrogen. Now, if you grow grapes, you probably know that there's not a lot of nitrogen, right, in the soil typically as compared to other cropping systems. So grapes are not a big concern for this type of emissions, right? But it can happen. It can happen after a fertigation event if you're applying ammonium or nitrate through the drip and with water, right? So, but it's usually pretty minimal. So these are something to take into account when we think about the role of soils in mitigating climate change. Now, soils are also important in the adaptation piece, climate change adaptation. And this is because they have a central role in the water cycle. The way we manage soils is going to influence how water gets into the soil, so the capture of storm water, how water moves through the soil, what we call percolation. And so that's relevant for the recharge of the aquifers. It also affects how water is retained in the soil, so water holding capacity. Even the interactions that plant can establish with microorganisms that will help them get water. For example, mycorrhizal fungi are very well known to help the plant take up water and nutrients from the soil. So managing the soil, and especially managing the structure of the soil, so the amount of pores, the aggregates in the soil, it's going to have a very strong influence on the capacity of vineyards and agricultural systems to adapt to droughts and also to floods, right? Because a healthy soil will allow the water to infiltrate better, right? Capture the rainwater better. And as I mentioned, all of that is because of the structure of the soil. And so the structure is the way the particles, the inorganic particles, sand, silt, clay, also organic matter bind to each other, and they generate the porosity of the soil. So this is what we typically see in a healthy topsoil, typical cookie crumb structure, you know, with the little aggregates, right? Those aggregates are what's going to hold the water. Those aggregates are important for water holding capacity because they have small pores inside. But also they create cracks in between them. Those cracks are macropores that are very important for water movement through the soil, infiltration, right? And percolation. So if we manage the soil right, this is what we see. If we don't manage it right, we could see this, which leads to a complete break of that structure and disintegration of the aggregates. So what we see there is soil that has been, the particles have been disaggregated, and it generates that sealing, right? With no pores, and that reduces water infiltration, and then, you know, that has consequences as well. It increases runoff and erosion, and you end up losing your topsoil. Okay. So then let's think about vineyards. What can we do to capture carbon and improve all of these physical properties that will help with climate change adaptation and going through droughts and floods? Well, a really good strategy to put carbon in the soil is cover cropping, right? No surprise. But the way we manage the cover crop, termination, whether you incorporate it or not, will drive the benefits of the cover crop, how much carbon stays, like the effects on the structure of the soil, and that drives the effects on water cycling. So what are the options? You can till, you can mow the cover crop and till it, right? And that can allow for a fast incorporation of the cover crop biomass in the soil. And some papers say that that could actually increase the carbon content, because you increase the contact with the minerals, right? So it could lead to higher increases of carbon, or at least faster increases of carbon. but you're also aerating the soil, and that brings more oxygen, and that improves decomposition and potentially losses of carbon through emissions of CO2. When you till the soil, if you go too far, you do it too intensively, you can break the structure of the soil, which leads to compaction if you drive the tractor a lot, which can lead to reduction in infiltration and, you know, higher rates of erosion and lower rates of water holding capacity. So everything is going to be connected to that structure. So the alternative is to no-till. Mow and leave the cover crop on top of the soil, right? And so in theory, that protects the structure. If you don't have a lot of passes with the tractor, then you don't compact the soil. The structure is going to be protected, which could lead to more carbon. It could also increase water infiltration and retention. If you have a living root in there, that also creates macropores. But some papers show that because now you have better structure, you could also have more anaerobic conditions in the soil that could increase nitrous oxide emissions, lower oxygen potential. So that has been shown in the literature. And some papers also show short-term decreases in yields because you're changing the way things happen in the soil, carbon and nitrogen cycle, right? And that could drive change in yields. All right, so lots of questions and unknowns, right? So the work I'm gonna show right now is 3 different projects with all this background that we just got, that it's trying to address the question of like whether no-till is a valid strategy for climate change mitigation and adaptation, and whether it has any effects on yields and agronomic outcomes. And we're doing this in 2 ways, following 2 approaches. The first one is to use control on-farm experiments, which is the best way to just separate the effects of tillage from other practices. And the second approach is landscape surveys, which is just going out to a bunch of farmer fields that are doing till or no-till, get samples, analyze them, and see how they compare. And so this is what I'm gonna show you. I'm gonna start first with a controlled experiment that we did. We started in 2018. This was down in the Central Coast in Paso Robles, Tablas Creek. And we got support from the CDFA to work as this was one of the first Healthy Soils projects, one of the first funding rounds. So we established this experiment where we had alternating blocks of no-till here with the dashed lines and till. That's where blocks of 4 lines of vines. And so from 2018 to 2020, those blocks were completely blocked off and there were no till, and the till blocks were tilled twice a year. There was a Syrah block with pretty high levels of organic matter already. And what we did over those 3 years is collect gas samples from 2 locations, the middle of the rows, what we call the tractor row, and then under the vines, we call that the vine row. So we use static chambers, and this is basically like a bucket that you put on the ground. It traps the gas that comes out, and then with a syringe, you take it and you bring it to the lab and you analyze it in a fancy instrument it's a gas chromatograph. So that's what we did over 3 years. Every time there was a management event or a rain event, we would go out to the field and for like 3 or 4 days in a row, collect gas samples. So we could see those flaxes of the CO2 and the nitrous oxide coming out. We got soil samples, and then we also harvested every year, and we measured yields, and we measured the quality of the fruit.
What was the difference in the in-row and the between row tillage, or was it not?
So tillage was done mostly on the tractor row.
Okay.
Under the vine, there was water. So that was mostly, but you see that this vineyard is kind of different because they have cover crops growing pretty much everywhere. Right, so they weren't doing like herbicide applications or anything like that. But yeah, water supply was the main difference. and because undervine it gets water during the growing season, the tractor row doesn't. Which could drive more N2O if you think about like what drives the nitrous oxide emissions being water, right? So we would expect that there's more nitrous oxide coming out in the undervine than in the tractor row.
In the vine road, is like put the gadget between emitters.
Yes, it wasn't right under, it was in between emitters, yeah, yeah. And also something that happens typically is that the tractor rows, because this is where the cover crop is grown most of the times, usually they have higher levels of carbon and nitrogen across many vineyards, right? The tractor rows where we see the higher concentration of organic matter in the soil as well. So this is what we saw in terms of carbon levels after 3 years. And so we took soil samples. You see here an example. We split them into depths. So the top soil was the 0 to 6 inches, in centimeters at 0 to 15. And then the lower part of the soil was 6 inches to 12. And so we analyzed the carbon content after those 3 years in till plots in dark gray, and then no till on light gray. So what you see here is like a very strong contrast between the topsoil, the lower layer, very typical. The topsoil has more carbon. We could expect that, but no differences in terms of treatments. So the tillage didn't really change the carbon much, right? So that was kind of frustrating.
Do you think it was just because it was such a short-term project?
Yes.
Yeah, it's a 1 year thing.
3 years. 3 years. But so this is one of the limitations of these studies. They are very valuable because, you know, we can control what's going on in the field and separate very well the effects of tillage versus no-till. But they only last 3 years. And the reason for that is funding, because these grants, they last 3 years. So, you know, at that time, it's like, okay, we have to finish. And it is a bit frustrating because then, you know, you don't really see much. And from what we know in soil science, it takes a long time to build carbon and to change things, right? That process of stabilization of the carbon in the soil, it takes a while. And this is not unique to this project, what we've seen here. We've seen this in like a lot of other projects where we just measure things for 3 years. Yes.
So what was in other words, what was the cover crop? What was growing in the no-till and what was growing in?
It was the same mix. It was a mix of legumes and grasses and yeah. We measure biomass in the two as well, no differences. And yeah. Why did you ask?
Oh, I was just wondering, there's a wide, there's a wide variety of
Yeah.
cover crops, and some of these, some of the ones that people use that they till under every year are sort of designed, or the idea is to increase organic matter. They're very large, some of these very tall
Yeah.
cover crops with like beans and peas and oats
Yeah.
and barley. So this wasn't that type of cover crop, I don't think, was it? Something
Yeah.
lower.
No, it was a mix, and in this specific vineyard, they have sheep as well. So I think they selected the maize for that as well. So it could also serve as feed. It was, yeah.
Yeah, but
But
you're,
you have a very important point that the choice of cover crop is important, right, for the purpose. Yeah. There was quite a, I would say, significant growth of the cover crop. So I would expect, that. Another reason why we probably didn't see change is that this was a vineyard that already had a lot of organic matter. So maybe it takes even more to see a change, right? And yeah.
So my first question is, clearly there is a little bit of a difference, but you're saying that the differences are not statistically significant.
Not statistically significant, no.
My next question is, why, I'm assuming there's a reason why you did not take a sample deeper than 12 inches.
Yes, because it was way more work. But we have other projects
But you
now.
think that the trends would show up
Yeah.
in the first 12 inches anyway, basically?
So most of the studies look at the topsoil, because that's where most of the carbon is, most of the biology is there. And so the changes should be visible in that topsoil. But you have a very good point. Carbon moves through the soil profile, and now there's also a lot of interest in looking at things beyond the first 6 inches, deeper than that. Because you could have actually a lot of carbon stored in the lower part of the soil profile, and that's where actually you have more of the minerals. So more potential for stabilization of the carbon and sequestration. So not in this project, we did learn a lot from this project about the things we didn't do. And so in the other projects we have going on right now, we are looking at down to a meter.
Okay, and I have one last question, which is, the last, lecture I attended about this
Yeah.
topic. Looked at a study that was in Oregon, and it was a 5-year study. And it ended because it ran out of funding. So, and it was a private vineyard. So my question, I guess, is why are these studies not being conducted in university-owned properties where you can conduct a study
Yeah.
over 25 years and see a long-term effect? Is it just because it's not a priority of the university?
I have the same question. The university should be for that, I agree. And there was a long-term study at Davis, the Century Experiment, was supposed to be run for 100 years. It was cut at 25. I think it was 2 years ago, they shut it down because of funding. So, yeah, so there's a lot of us faculty that are kind of trying to push for for these things to be kept, because there's a lot of value, beyond the economics, of like keeping these trials. And yeah, maybe at some point we have it.
But there's that study that they did in Switzerland and Germany that went for a very long time, like
Yes,
the
in Europe, we
artificial,
have done quite a lot of work.
the artificial, what would it be if the CO2?
Yeah.
Right, that there were collaborators at Davis with that as well,
Yeah.
that many of them are retired or gone now, but.
Yeah.
So there, it exists, just
There-
not, it's
yeah.
dollars, that's.
Yes, it's keeping those trials, running. It takes time, right? And what happens is also like, you see a lot of change in trends, like as well, so it's kind of, farming industry kind of also moves, like from like drip irrigation now, everybody uses drip irrigation, right? So, but it wasn't a thing maybe 15 years ago, so things kind of change, so keeping those long-term experiments is also a bit of challenging in that sense. But I agree that the university would be a perfect place for it.
If you went back 10 years to this same vineyard, would
Yeah.
you see something different than the two bars? that
Okay,
you've seen?
so this is something I'm not showing here, but we did see things changing in the soil, right? Now I'm focusing on the total carbon, which is what most agencies look at, like the CDFA just wants you to report total carbon as a kind of a show that you are changing things. But you can change things in the soil that are not total carbon that will show a trend towards carbon sequestration. Okay, things that show that changes in microbial biomass, right? If you have higher microbial biomass, you might not increase your total carbon, but you're increasing the growth of microbes. And I mentioned before that that's one of the pathways, right? So that would be a good sign. A very good indicator for the short term, it's active carbon. Have you heard of that?
Yeah, the permanganate test.
Exactly. So POC, they call it, or permanganate oxidizable carbon. It's a fraction of the total carbon that is actively used by microbes, contributes to microbial growth. Most of these studies don't show changes in total carbon, but they show changes in active carbon, which shows that there's more feed for microbes, therefore there could be more microbial growth, therefore more carbon in the future. So these are all signals that if the practices are maintained, then you will see a change. So there are early indicators. Another thing we've seen also in this study, changes in the microbiome. That happens immediately, especially with tillage. Fungal populations, completely different. There's a paper coming out actually now that will show that. The microbial community is different in a till and a no-till soil. The fungal population is affected the most because tillage disrupts the fungal hyphae, right? So the growth of the fungi is going to be very directly affected by that. And fungi have a very important role in carbon sequestration, right? So the ecosystem starts to shift. And this is what we need to also take into account. We're shifting the soil system. We might not be changing the carbon, but we're shifting the soil system in that direction. So that's something also probably to tell the funding agencies, maybe to move away from the focus on carbon and to start thinking about other indicators, early indicators of change in the soil.
Talking about tilling, how deep are they tilling?
This was kind of like a surface tillage, I think, a few inches.
Just a few inches?
Yeah, not very deep.
Not rip.
No, yeah, and that's another thing, like, there's so many types. I'm gonna go on because I want to make sure I cover everything. All right, so this is the emissions of the gases from the beginning in 2018 to 2020. So you see, we talk a bunch of different measurements. And what you see here, this is CO2 for the vine row, and the top graph is the tractor row. The arrows show the tillage events during the experiment. We kind of split it into dry and wet seasons rather than years, because that's what drives the emissions, the levels of moisture in the soil. So dries were from April to October, and then wets were like basically the dormant season. So what we've seen is that CO2 emissions increase during the wet seasons, which was expected. There's more moisture in the soil, plants are growing, microbes are active, and also during the first dry and wet season, during that first year of study, higher emissions of CO2 in till plots as compared to no-till plots. But then the trend was reversed on the following wet season. So we saw higher emissions on the no-till plots. So, things could go both directions. If we think about the nitrous oxide, so this is the same type of graph, emissions through the experiment and the tractor row and the vine row. No differences whatsoever. Good news. So we're not really increasing or decreasing N2O. Emissions are minimal, really small compared to other cropping systems. So this is not a concern in vineyards in general. Not a problem you have to worry about. We looked at soil physical properties to see, if we were potentially changing something with water cycling. So this is the tractor row, the vine row, the two different depths and the no-till and the till plots. We look at bulk density, stable aggregates, the percentage of stable aggregates, and water holding capacity. So how much water is the soil holding? Main message, no differences whatsoever in bulk density, water holding capacity. We did see differences in stable aggregates, but only between soil depths. So the topsoil has more stable aggregates than the bottom part of the soil. So also, kind of expected, but no tillage effects in these soil physical properties. And then when we look at yields and quality, so this is the yield of the crop in 2018, 2019, 2020. This is anthocyanins, this is phenolics. You can see lots of variability between years, but this is the no-till and the till treatments in gray and orange, which were very similar each year, right? So besides that interannual variability, tillage didn't really change anything. The yield and the quality was similar. Okay, so to till or not to till? 3 years after, they implemented the practice, they removed tillage from this vineyard. Nothing really changed much. There were no clear benefits for climate change mitigation or adaptation, so there was no more carbon or nitrogen in the soil. Slightly reduced CO2 emissions in the no-till as compared to the till system. But overall, if you put all of the emissions together, cumulative emissions were different between the till and no-till. And there were no changes in soil physical properties either that could change water cycling. So things were kind of the same. But on the other hand, if you think about it, it's like there were no trade-offs. So this farmer could stop tilling and, use less gas. There were no higher N2O emissions, and the yields and the quality were similar, right? So there weren't any trade-offs as well. But overall, frustrating, right? It's like, okay, we see the system is changing. We don't see changes overall in total carbon. So this is when we started thinking about the second approach. Let's get out and find fields. We can't keep this experiment for more than 3 years. What about if we go to fields, vineyards, where they have been doing this for more than 3 years? We get soil samples and we compare them across the landscape. So this is the work of the PhD student in my lab, Noelimar González Maldonado, which some of you may know. I think she presented in this group a few years ago. So her PhD project is looking at soil health across different vineyards in Napa. And she's kind of looking at soil health indicators. So the scope of her work is bigger, but she went out to 32 vineyards. And it turned out that out of those 32 vineyards, 13 were this, so till, they had a certain level of tillage, and then 19 were no till. And so we said, well, let's see what happens if we compare those. And so we look at all of those different soil health indicators, and we put side by side the disc soils and the no-till soil. And what we've seen is that no-till increase, for example, VOC, which is dissolved organic carbon, is a form of carbon that is available for microbes. It increased soil pH across all of these different fields and increased electrical conductivity slightly. But on the other hand, the soils that were no-till also had lower levels of POC, active carbon, lower levels of total nitrogen, and lower levels of nitrate as well. And there were no effects on total carbon, aggregate stability, and water infiltration rates. So looking at things across the landscape, it's also quite complicated, and you probably know why. Because there's so many confounding factors, right? Like, okay, when you look at things beyond 3 years, if you want to see what happens if you implement the practices in the long term, it seems like tillage is driving the way carbon and nitrogen is being cycled, but it's really hard to see trends across the landscape because of these confounding factors. Among many other things, there's what we call static soil properties like texture, even like landscape position and slope, that drive soil health and the response of the soil to the practices. And then many times, no-till is combined with like many other practices as well, right? Like the type of cover crop that you choose. Some people are doing grazing, right? Or like even the tillage implement that people use or how deep they till. So this is also not an easy way of looking at things. So then we follow yet another approach where we're kind of like trying to see this idea of like stacking practices on top of each other, right? What if you start adding, no-till and cover cropping and compost, and also you add sheep grazing, which is what a lot of growers are doing, right? They use a different combination. And so that's what we're doing in this project, that it's ongoing. So the data we have is still preliminary, but so we're trying to kind of look at this stacking of practices and whether a site that has minimal soil conservation practices, like in this area, everybody uses cover crops, so most of the people have cover crops. But then we also compare it to growers that are using cover crops and no-till and compost and grazing, and see how that compares. And we also wanted to do it in the same soil, right? Because that matters. And so what we did is compare paired sides. So vineyards that were like in the same type of soil side by side, but they were using these different sets of practices. So we had 3 paired sides. The first one, what we call the soil health side, had no-till, cover crop, sheep grazing for more than 5 years. Then the conventional had tillage and disking and bare floor and no grazing. Something similar for the second pair side, no-till for more than 34 years, cover cropping, compost, biochar, sheep grazing, and the conventional was tillage and disking, no grazing. So you get the idea, right? So this was done in Napa mostly. And here are the results. This project specifically focuses on water, soil physical properties that will lead to more water retention and infiltration. And one of the first things that we looked at is the proportion of stable aggregates, the structure of the soil. And so here you have the 3 paired sides in these columns. And then the conventional management is in red, then the soil health management is in blue. So across the 3 paired sides, higher aggregate stability in the soil health field as compared to the convention, right? One of the main differences here was the use of tillage, but there's also stacked practices. With these values of aggregate stability, we calculated an erodibility factor, which is how likely is that soil to be disrupted by rain, right? And to be eroded. And so we see the opposite trend. Now the conventional soils have a higher likelihood of runoff and erosion and than the soil health sites. Another thing we did is looking at the size of the aggregates, because that matters, right? Like you want to have aggregates that are stable, and also that the size of those aggregates will tell you a little bit the size of the pores and therefore how much water you can retain and infiltrate, etc. So here you have the 3 sides stacked on top of each other, and then from left to right, you have like from macro aggregates, so the larger size, you have there 8 millimeters to the lower sizes here, and this is mostly silt and clay particles. So that this will be disaggregated minerals already, okay? So if you look at the 2 different colors, you'll see the trend there too, right? Focus on this side of the graph, right? What do you see there?
Higher, bigger.
Larger aggregates in the soil health sites, right? As compared to and the opposite happens if we look at the other end of the spectrum. The conventional sites have higher proportion of those disgregated mineral particles, clay and silt, right? So this is telling us that soil health practices lead to higher porosity, and potentially that could affect water retention and infiltration in the soil. And so this is what we're working on right now. This is a process, it takes some time. You need to kind of subject the soil samples to pressure, and it takes a few weeks, and we have been collecting samples until yesterday in the field, so it's still kind of ongoing. But we're looking at how much water can the soils retain, right? This is important because you've probably seen this number thrown around a lot of times, like for every 1% of increase in organic matter, you can have 20,000 gallons of water more. Well, this is a number that was seen maybe in one paper in the Midwest, but it's definitely probably doesn't apply for vineyards in California, right? So we need to know whether we have a benefit from increasing carbon and increasing organic matter for water, right? So this is what we're doing at the moment. And we hope to have data early next year. And I'm wrapping up. I know I'm going a little bit over time. I also wanted to mention another project I'm working on in collaboration with Dr. Beth Forrestal, which you may know from UC Davis as well. So we're looking at how tillage and cover crop management helps vine and vineyards be more resilient to heat waves. And I know this is something that many of you are worried about. I heard of a lot of growers that are already measuring temperature in the vineyard floor and they're seeing differences. If they have cover crop, which is mulch on the soil, there is potential for decreasing the floor temperature, the soil temperature, and changing the microclimate in the vineyard. And that can affect how the vines respond to that heat wave. And that could change impacts of the heat wave in grape quality. And it could also help the microbiome in the soil and how the microbiome is working and doing their job, sequestering carbon, cycling the nutrients, etc. So this is something we want to look at. We have also one experiment that we started, and we hope to expand in the next years. If you're interested, reach out, because now is the moment when we're talking to growers and kind of like gathering collaborators. And so, yeah. Okay. And that's it from my side. And if you have questions, we can revisit any of these graphs that I just showed you. Yes.
Different soil types, we have a soil type that's high in adobe content and it cracks a lot. And we've tried, a few years ago, we tried no tillage and there'd be huge cracks, and then the moisture, we'd lose moisture. Basically, we had to irrigate more, so we've gone back to tillage.
Yeah.
Does that sound?
So, like heavy clay soils are really hard to manage under no-till. So like going no-till is definitely quite the challenge most of the times. And so maybe in that case, it's like strategic tillage and strategic disturbance. And if you need to do it, then yes. The only thing I would recommend is if you're interested in soil health and also water retention, just look at the changes, with the tillage that you're doing. Maybe get a few samples, every year, and then see, how things are going. If your carbon is changing over time, and yeah.
At the same time, we had a neighbor that continued on with that no tillage, and his yields went down, his vines decreased.
With the no-till?
Yes,
Yeah. Okay, that might be a place to start to study. Yeah.
Sure. It's great. It's good work you're doing.
Yeah.
Vineyards are temporarily permanent crops. So say we establish a no-till vineyard and we have it there for 30 years, and then we do our typical redevelopment protocols, ripping, cultivating, things like that. What effect does that have on built up, microbiological, flora and fauna and things like that?
Yeah,
And soil
vineyard establishment. From what I know, what I read, like establishment, especially the first time, has the strongest impact of everything you can do in a vineyard, even like afterwards. It's that change, the establishment of the vineyard, the change from like natural to a cultivated land.
Or even if it's vineyard that's been no-till for 30 years and then you redevelop to another vineyard.
It also has a pretty strong impact. Yeah.
Yeah, it'll take years to rebuild that structure and redevelop that soil health. We're in the middle of that right now. We planted our whole 10 acres over the last 5 years in 3 stages.
Yeah.
And it was such healthy soil before, and
then it's
it's just dust now.
wow.
Take us years to build it back up.
Did you rip?
Did you rip? Oh, yeah. Rip, and we're in a really cool climate where it's particularly hostile for nurturing young vines, where we really, any competition at all makes it really hard. So we've just been,
It might go faster than you think.
I hope you're right. I hope it does.
That's my experience.
Yeah, I mean, once we get cover crop in this winter, things will be, you know,
If everything was, good to go beforehand and you did, kind of common sense.
good. Right on, I like that. Activity, optimism.
Yeah, I guess that's something also, it's true that happens, and there were some long-term studies not on vineyards, but tomatoes, Central Valley, 20 years of no-till. And, they saw benefits as soon as they stopped, gone. So, yeah, that was work that I think Jeff Mitchell had done. And so it's important also to understand that it's constant work. It's like if you stop going to the gym, then. Yeah.
What the underlying vegetation management practices were at Tablas Creek, were they mowing, were they weed eating, are they using an under the vine mower? Cause it looked pretty clean.
They were mowing, yeah.
But they were, what, do you know actually what they were mowing with?
I can't remember if it was like I can't remember what implement they used. Yeah. I can look it up. Yes.
I could call.
When we're using strip spray or herbicide management under vines, you want to focus on, build that soil health in that strip spray, whether organic or non-organic. Are there any materials that we can add through the drip that are gonna help to increase, bacteria, fungi development there so that we can affect soil health, or is that pretty much just a feel good thing?
Well, I haven't done it myself, but I've seen studies from collaborators at Cal Poly that they use liquid vermicompost, and it worked pretty good to change the microbial community. Yeah. So they saw an effect pretty quick.
What did they add?
Liquid vermicompost.
Looks like a compost tea.
It's like a compost tea. Yeah. So what you do with that is like you add like maybe more easily available carbon for the microbes. So it has a pretty quick effect. It's nutrients and it's dissolved carbon, which is going to feed the soil microbiome. Yeah. It works. Other things I don't know about, like I know there are other products, inoculants and that. I don't know about those ones, but like compost is like I would say a pretty safe bet. If it's dissolved, you can put it in the vine and it has clear effects. Okay.
Questions? If not, I want to thank you, Cristina.
Yeah,
again
thank
for
you.
everything. Thanks, everyone. Good luck to harvest. Yeah.
Yeah, good luck with harvest.
Get through the rain. 60% of your neighborhood. 80, yeah.
Why don't you grow?
There weren't any questions. Okay. You want to go with us to lunch?
Sure, are you buying, Jason? You, but I don't have the time.